Like the eukaryotic cells, bacterial cells also possess an ultra-thin, elastic and living membrane known as plasma membrane. Plasma membrane is absolutely required for all living organisms. This membrane protects and separates the cytoplasm from the external environment. This membrane is considered as an highly organized, asymmetric, dynamic and flexible structure that perform many roles which are vital to the organism. Hello friends, welcome to some biology class. Friends, today we shall discuss about bacterial membranes. First, bacterial plasma membrane. Bacterial plasma membrane is composed of roughly equal amount of proteins and lipids. Most of the membrane associated lipids are phospholipids. They are amphipathic molecules having both polar and non-polar end. The polar end is water soluble and is hydrophilic, and the non-polar end is insoluble in water and is hydrophobic. Such amphipathic lipids in aqueous solution form a lipid bilayer in which the polar hydrophilic head group remain exposed toward the aqueous environment and the nonpolar tail remain buried in the interior of the bilayer. Under the transmission electron microscope, the plasma membrane appears as a thin structure having thickness from 5 to 10 nanometer. It appears as two dark lines separated by a light interior. The structure of plasma membrane is best explained by fluid mosaic model which was proposed by Singer and Nicholson. According to fluid mosaic model, the plasma membrane is composed of a lipid bilayer in which these protein molecules float. Here you can see that this is a lipid bilayer. There are two layers of lipid. This is the polar hydrophilic head group and this is the non-polar hydrophobic tail. In such a lipid bilayer, the polar hydrophilic head group remain exposed toward the aqueous environment and the non-polar tail remain buried in the interior of the bilayer. And these are the protein molecules that are associated with the membrane. These protein molecules can be divided into two different groups: peripheral membrane protein and integral membrane protein. Peripheral membrane protein remain loosely connected with the surface, thereby they can be easily removed. These peripheral membrane proteins are soluble in water. And other group of protein is integral membrane protein. This integral membrane protein cross from one side of the plasma membrane to the other side. Like the phospholipids, these integral membrane proteins are also amphipathic. Their hydrophobic region remain buried in the lipid bilayer, whereas their hydrophilic portion remain exposed to the aqueous environment. In the plasma membrane, here and there you can also see carbohydrate groups that remain attached mainly on the outer side with the lipid as well as with the protein where they serve important functions. One of the distinctive features of bacterial plasma membrane is the absence of sterols such as cholesterol. Sterols are present in eukaryotic membranes. However, in bacterial membranes, sterol-like molecules known as hopanoids are also present. Like sterol, hopanoids probably help in stabilizing the membrane. So friends, this is about the structure of bacterial plasma membrane. Now we shall discuss about the structure of Archaeal membrane. Archaea is a group of most primitive prokaryotic organism. The basic design of archaeal membrane is similar to that of eukarya and bacteria. The archaeal membrane also has two hydrophilic surface and a hydrophobic core. But the nature of the lipids that are present in the archaeal membrane are very much different from that of eukarya and bacteria. One of the most distinctive features of archaea is the nature of their membrane lipids, which differ from that of eukaryote and bacteria in two ways. First, the membrane lipids in archaea contain hydrocarbons which are derived from 5 carbon isoprene units. Thus, the hydrocarbons are branched. Second, these hydrocarbons are attached with the glycerol moiety by means of ether linkage rather than ester linkage. When two hydrocarbons are connected with a glycerol molecule, diether lipids are formed. These are the diether lipid molecules. Diether means a glycerol molecule is connected with two hydrocarbons by means of ether linkages. And the diether hydrocarbon chain usually contain 20 carbon atoms. But sometimes tetraether lipids are formed when a long hydrocarbon chain containing about 40 carbon atoms link two glycerol molecules. This is a tetraether lipids. These are the two glycerol molecules which are linked by two hydrocarbon chains which contain 40 carbons. The cells adjust the overall length of tetraethers by cyclizing the chains to form pentacyclic rings. In addition to this, the third carbon of glycerol is also connected with phosphate, sugar and sulfur containing groups, thereby making the membrane lipids polar in nature. Usually 70 to 93% of membrane lipids in archaea are polar lipids. Remaining lipids are non-polar and they are derived from squalene. Despite of all these significant differences, the basic design of archaeal membrane is similar to that of eukaryote and bacteria. There are two hydrophilic surface and a hydrophobic core. When the membrane is composed of C20 diethers, a regular bilayer membrane is formed. But when the membrane is composed of C40 tetraethers, a monolayer membrane is formed which has much more rigidity. These two figures shows the structure of archaeal membrane. In some archaea, the plasma membrane is composed of two layers of C20 diethers in which protein molecules are embedded. This is the integral membrane protein, and this is a hydrophilic surface, and this is another hydrophilic surface. These are the polar hydrophilic head groups. And these are the hydrocarbon chains which are derived from isoprene subunits. But in some other archaea, for example in case of thermoplasma and sulfolobus, which grow best at 85 degrees centigrade, the plasma membrane is composed of a monolayer of C40 tetraether lipids. These are the molecules of C4 tetraether lipids in which two glycerol molecules are linked by two long hydrocarbon chains having about 40 carbon atoms. This is about archaeal membrane. Functions of plasma membrane. Plasma membrane is absolutely required for all living organisms because of which cells interact with their environment in a selective fashion, acquire nutrients and eliminate waste. This membrane also maintains a constant internal environment. Plasma membrane is a selectively permeable barrier. It allows the movement of specific ions and molecules into and out of the cell at the same time, prevent the movement of others. It prevents the leakage of essential constituents. It is the location of various transport systems. As you know, in bacteria, membrane-bound organelles are absent, so the plasma membrane is concerned with various vital metabolic processes. For example, energy generating function. Bacterial plasma membrane is the location of respiratory and photosynthetic electron transport. Across bacterial plasma membrane, protomotive force is generated. It also contains the transmembrane FO-F1 ATPase, which is responsible for the synthesis of ATP. The bacterial plasma membrane is also the site of synthesis of phospholipids and peptidoglycans. It is concerned with the assembly and secretion of extracytoplasmic proteins. It is the site of insertion of flagella, and finally, the bacterial plasma membrane also contains special receptor molecules because of which bacteria can detect and respond to chemicals in their environment. So friends, this is all about structure and function of bacterial plasma membrane. For more updates, please subscribe Sam Biology class and also please share your comments. Thank you very much. Good night.